Hi, my name is Evan Fowler. I'm a junior at the University of Alabama in Huntsville. I had the privilege this summer of working with Dr. Sharifa Love-Rutledge in order to analyze type 1 diabetes in a rodent model. We found that Lewis 1WR1 rats have increased beta cell area and decreased pancreas area during their type 1 diabetes susceptibility window. So why are we doing this research in the first place? Well, to understand diabetes, you first have to understand a little bit about anatomy and physiology. On the left picture, that shows a diagram of the pancreas. It's the hot dog-shaped organ sitting right below the liver. On the right photo, you'll see that there are clusters of cells in the pancreas called islets of Langerhans. And these islets contain what's called beta cells. And the beta cells are the cells that secrete insulin. Now, why is insulin so important? Well, when you eat food, when you eat a meal, your stomach breaks down that meal into its monomers, amino acids, carbohydrates, and lipids, and this, among other things, causes a spike in blood sugar, in glucose in your blood. This signals the beta cells in the pancreas to start releasing insulin. Now, as insulin starts traveling through the circulatory system, it interacts with receptors on the surface of blood cells. And it starts a biochemical pathway in those cells that causes a membrane transport protein to start being produced and placed in the cell membrane of those cells. This transport protein is a channel that allows glucose to cross the cell membrane and go into those cells. So from a big picture point of view, when you eat a meal, your blood sugar increases. This signals the pancreas to release insulin, and that insulin is the signal for your cells to take up that glucose and bring your blood sugar down. Diabetes occurs when the beta cells in the pancreas can't secrete insulin like they normally would, and this leads to blood sugar problems. There are two major types of diabetes, type 1 and type 2. Type 2 diabetes has genetic components, but it's also a lifestyle issue. If you eat just too many calories, or maybe you eat the number of calories you need, but you just eat a lot of sugar, over the period of a few years, this will stress the beta cells in the pancreas and cause them to undergo apoptosis or programmed cell death. And once those beta cells are dead, you're not going to be secreting insulin like you once were, which leads to blood sugar problems. Type 1 diabetes is often diagnosed in children because it's not as much of a lifestyle issue. For some reason, the immune system, which normally attacks or destroys pathogens, attacks and destroys the beta cells in the islet of Langerhans. And this causes impaired insulin secretion. In both cases, insulin's not being secreted, and therefore blood sugar remains high. And this leads to all kinds of health problems such as neuropathy, maybe even blindness, infections, which can lead to even amputation of limbs if left untreated. So diabetes is a serious disease. Now, our lab is searching for a biological test for type 1 diabetes susceptibility. And why are we doing that? Well, the idea is that one day, if we have this test, medical professionals will be able to intervene in patients before they get to the point that their beta cells are destroyed. You see, right now in healthcare, a patient comes in, maybe a child who is a type 1 diabetic, undiagnosed, and the child says that they are urinating incredibly frequently, they're incredibly thirsty, and they just feel lousy because their blood sugar is high. By the time you see those symptoms, the beta cells have already been destroyed. So if you had a biological marker for type 1 diabetes susceptibility, this will help medical care hopefully progress to the point that we're trying to prevent this disease rather than just treat the symptoms. The analysis that I did actually was sort of a spin-off from a previous analysis of data done on a previous study. So the actual live experimentation on the rats was done before I joined the lab back in 2018. There were two strains of rats used, Lewis 1WR1 rats and Lewis SS and HSD rats. The 1WR1 groups were susceptible to type 1 diabetes induction. This is due to the fact that their FAT10 gene is defective, and this leads to modulation in diubiquitin expression. Now, poly-IC is the actual mechanism used to induce type 1 diabetes in these rats. And poly-IC is just a synthetic version of double-stranded RNA. So when poly-IC is injected into these rats, it stimulates their immune system, which causes the immune system to attack the beta cells in the pancreas and destroy them, and that induces type 1 diabetes. So the 1WR1 rats modeled type 1 diabetes susceptibility, and the SSNH-SD rats modeled type 1 diabetes resistance. And then for each strain, you had a treated group that was treated with poly-IC and a control group that was just given essentially IV fluids. Now, when a previous researcher analyzed data from this study, she found that the islet number for the 1WR1 groups were significantly lower than for the SSNH-SD rats. So my analysis was to check and see if there were any sort of correlations between islet number and pancreas area and islet of Langerhans area. So how was the experiment done? Well, like I just explained, we had 4 groups. Each strain of rat had 2 groups spanning from it. You had a group for each strain that was treated with poly-IC and a group for each strain that was untreated. Each group received injections every other day for 6 days. The poly-IC group received poly-IC injections. The control groups received phosphate-buffered saline, which is essentially IV fluids, so it wouldn't really change our results. After the 6 days, they were placed in metabolic cages, and in 7 hours, the animals were sacrificed. Their pancreases were fixed in formalin and sent off to Histowiz. Histowiz is a company in New York that takes tissue samples, puts them on slides, and digitizes them so that researchers can sit in the comfort of their offices with their computers and zoom in and look at the different cells on these slides. So this was very convenient. All the pancreases I looked at were on the internet. I took these images from Histowiz. I took islet of Langerhans images and pancreas images, and I opened them in ImageJ, which is an open source software from NIH. That allowed me to find the area of the islets and the pancreases. Once I got those numbers, I analyzed them on GraphPad Prism. I started out with an analysis of variance, ANOVA, and that told me out of the groups of data for the different rat groups, were there any differences that were worth noting? Were there any statistically significant differences? Once I did that, I did a Tukey test to compare individual means. So a Tukey test allowed me to take each individual rat group and compare it to the averages of every other rat group to see where the difference was coming from. This is what it looked like while I was gathering samples from Histowiz. The white box with the purple shape in it towards the lower left corner of the web browser screen, that's a total pancreas. And the tiny red box in that little window was my viewing window. You can zoom in and see individual cells. It's very nice. The brown you're seeing, those are beta cells. The brown color is where insulin was stained for. So I had to go through each pancreas and find a sampling of the islets. I didn't go and get every single islet. I just got a representative sample of the islets on that pancreas. And I also got total pancreas shots. Once I got those screenshots from Histowiz, I opened them in ImageJ and measured them. This photo is an example of me measuring total pancreas area. Now, I didn't have to trace every single portion of this image. Some of the pieces of the image towards the right that are lighter colored are gut tissues or connective tissues. So I had to discern between what was actually pancreas and what was just extra tissue thrown in there. Once I traced them, ImageJ had a measure function that gave you the area of the image in pixels. I didn't want the area in pixels, though. I needed the area in the actual millimeter square measurements of the pancreas. So I had to convert it. So how do we make this conversion into numbers we could actually use? Well, I measured the scale bar included with each image, and I found the length of each pixel and how it corresponded to length in the actual pancreas or islet of Langerhans. I took that length and squared it to find the area of each pixel and therefore the area it corresponded to on each pancreas. And this gave a conversion factor that I could use to take those pixel area measurements and convert them to actual numbers that were usable. Once I did that, the top diagram, the pancreas area, was done. We had it in millimeters squared. Normalized beta cell area was a little bit different to come up with. So I had the area of all the islets for each pancreas in millimeters squared, but I had to do something else to normalize the data to prevent the results from being skewed. You'll notice if you look at the top graph that all the pancreases were different sizes. So that could introduce some bias into your study if you didn't take that into account. Pancreas could appear to have higher beta cell area just by virtue of the pancreas itself being a lot bigger. So to control for this, we took the total islet area for each pancreas and divided by the total pancreas area for that pancreas. And so normalized beta cell area is being reported here as a percentage of total pancreas area. So that gives an even comparison for each rat. And now what were our results? Well, as you can see in the bottom graph, the 1WR1 treated group had statistically significantly higher beta cell area than the SSNH-SD control group, which surprised me. When I was unblinded, I hypothesized that as the immune system were attacking those beta cells and they were decreasing in number, the beta cell area would go down. But the opposite happens, so that was surprising. I also didn't think that pancreas area would really be any different between the rat groups, but it was. As you can see in the top graph, the 1WR1 groups, both of them, had statistically significantly smaller pancreas area compared to the SSNH-SD treated rat group. And we were looking at the small lowercase letters in these graphs. The different levers represent statistically significant differences where P is less than 0.05. As far as the conclusions, I observed that Lewis 1WR1 rats had increased beta cell area and decreased pancreas area during their diabetes susceptibility window. This one analysis doesn't prove that islet area or pancreas area can be used as that biomarker I was discussing earlier in the presentation, but it does set it up for future studies. I'm going to be continuing that, whereas in this study I got a sampling of beta cell area in each pancreas. I'm currently working on going back through each pancreas and getting each individual islet and measuring it. So I'm going to see if the numbers that I'm gathering now match up with and show the same pattern that I've previously seen. And other members of the lab are evaluating other biomarkers for their potential as type 1 diabetes biomarkers. Here are my references. The first is a nice paper that explains why the 1WR1 rats are diabetes susceptible. Like I said, it has to do with diubiquitin, or UBD. The second is a presentation that the lab group gave before I joined, and that's where I got my first graph from, the one with islet number. So this analysis is the one that gave rise to my analysis. I would like to take a moment and say thank you to several people who have made this experience possible. I want to thank Dr. Bernhard Vogler and Mr. David Cook. They've done a great job setting up their RCU program, and it's been a wonderful experience. The Office of the Provost, the Vice President for Research and Economic Development, the Dean of the College of Science and of Engineering are partially funded this project. I appreciate them. ALSAMP, NASA, and the Alabama Space Grant Consortium, they participated in funding this project, and I appreciate them. And I'd also like to thank God for giving me the mind to even be able to go to college and give this presentation. So, lastly, I would like to thank you, the viewer, for giving your time to this presentation. I hope you've learned something.